So I'm really, really happy to be hosting Sriram Ramesh and Jorge Tapias Gomez to discuss the recent PLOS ONE Lab Protocol paper entitled Reference Sequence Browser and our application with a user-friendly GUI to rapidly query sequence databases and the associated detailed protocol in protocols.io. Jorge earned his bachelor's in science in computer science from the University of California in Santa Cruz. As an undergraduate, he joined a student-led research group where he learned about environmental DNA and used his coding skills to help develop a computational biology tool to aid scientists with their research studies. Since then, his focus has shifted and he is now pursuing a PhD in computer science at Cornell University, working in collaboration with the medical physics department at Memorial Sloan Kettering Cancer Center. His research is centered on developing machine learning methods to assist doctors in detecting and treating tumors. Sriram finished his undergrad in 2022 with a bachelor's in science in computer science from the University of California in Santa Cruz as well. Since then, he has been working for a supply chain startup, but is now taking a break to apply for grad school programs. His hope is to work more with the intersection between database theory and bioinformatics. Sriram, Jorge, thank you so much for carving some time to talk to me today.
Thank you for the great introduction.
Thank you for the thank you for the invite as well.
Yep.
So to kick off, can either of you explain what the protocol is about, why it is relevant, and how researchers use it?
Yeah, so basically, on a high level, kind of like overview, there's like our target audience, right, or like the field that we're, this tool kind of works in is called like eDNA, which stands for like environmental DNA, and metabarcoding, which basically, scientists, they go to the field, they collect some DNA samples, and then they come back to the lab, and then they compare these samples they collect of DNA to some references that they have, some reference sequences, we call them. And then they can detect what it is that they've collected, right? So if I go out in the field, I collect some DNA, say it's like a chicken, I have to go back to the lab and then compare it. And then if it matches, then that is a chicken, right? And so basically, before you can do any study in this kind of field, you need to collect this database of reference sequences to be able to, compare them against whatever you collect. And so to do this is kind of like a tedious process right now. It's a little bit manual. Or some people, automate it, but it's through their own, scripts and coding, and not everybody has that expertise. And so for a lot of people, this process can take, a few days to like a week in the worst cases that we've seen. And usually this process, basically what it, I guess like what it looks like is you go to a database, that could be NCBI or it could be Bolt, and then you kind of like search whatever you're interested in. So, Gallus, for example, with some barcode, and then you download all the sequences that you're interested in. And then if some database doesn't have it, you have to go to, search around a little more, right? So you might go to Bolt, or you might go to Crux. And so you kind of have to go through different databases one by one, and you have to search your species one by one. And this takes quite a while, right? So that's kind of like the idea of our protocol, and why we kind of created this tool, right, that automates this process. Instead of going one database at a time, you have this tool that has all the databases together, both NCBI, Bolt, and Crux, and you can then input your species and your barcodes that you're interested in, and it will automatically search these databases, and you can explore the results that you have, analyze them, and then download all those references. That's the gist of what this protocol encompasses, and what the field, where the gap in the field is.
And I would like to hear about how you came about this idea, like how the project started.
Yeah, so this started actually as out of a class that I had taken in undergrad in UC Santa Cruz, there was the class itself was more of about project development and a mix of that and some business, regarding finding product market fit, finding like uh, the way that the class was designed was that the students were grouped up into small groups, and each group was given some project field to explore and find problems in to solve. And basically at the end of the class, myself and some of the other students realized that like we had over the course of that class like found a problem that we thought was very solvable. Like this is this was an actionable thing that we could tackle. And so independently, we didn't really have any lab that we were working under or any strict mentors. We sort of just took this on as our own initiative to try and make this tool. And we started doing a lot of cold call interviews with scientists who are working in this field to make sure that we really understood the theory and that every step of development, the tool that we were making was something that they felt that they would really be helped by and that they would really use.
And as you were developing the protocol, what were the highs and lows? And was there anything that you tried to optimize that didn't work and didn't make it to the final version of the protocol?
Yeah, so kind of like as Sriram was saying, this all started as a student-run, like led group. So we don't really have a PI that would guide us through the process. And so there were, you could say lows, right? There were many lows where, we would encounter problems that, you don't see coming. Also, we were not familiar with the field at the time, right? We were computer scientists, and also we were undergrads at the time. So publishing papers, creating tools, these were all like very foreign concepts that are very different from a class where you just, create something and turn it in. And so, we had, I guess, some issues that we run into were, what we call feature creep, where we wanted to keep adding stuff. You want to keep analyzing data. You want to keep like helping the scientists by adding more stuff. But the problem is that the more you add, the more difficult that, maintaining the tool is. And also the more time you have to spend. So at some point we had to kind of like, cut back and say, okay, this is like enough for now. Let's, stop here, publish the paper and, get other people to use it. And then some of the highs were how much we learned throughout the project. I mean, like one of the advantages of a student-led and struggling all of this is that you learn a lot. We learned about publishing papers on our own. We learned about writing the paper, creating a tool that works as a web page, but also like downloadable, and also a lot of interviewing. It's not really grant applications, but we did a lot of like trying to get money to like publish this paper and pay for PLOS ONE. So, there was a lot of involved in this project, and so we learned a lot. And there were certainly features that we didn't add. There were many, for example, homonyms. There's something in like biology where it's not, sometimes there's like duplicates, I guess, like homonyms where two species have the same name. And so, what do you do when someone is trying to find Gallus gallus, but there's like two different types of Gallus gallus? And so we've tried to tackle that problem, but it's not like a feature that is included in every database yet. And so there's certain features we still haven't implemented that we want, but we haven't had the time. So there's definitely things we have cut back and, that we didn't have the time or the ability at times to actually do it. But yeah.
As researchers and users interact with the protocol and with the tool itself, what are the most critical steps, the most critical parts of the protocol and the tool itself to make it work successfully?
Definitely writing on what Jorge was mentioning earlier, because this tool, unlike some of the other tools that are available right now, this is using keyword searches. The users should just be a bit aware of the fact that the taxonomic tree is not constant. We learn new things about evolution and things get reclassified. So names can change a bit. And like Jorge mentioned earlier, there are the case of homonyms. This is more of a situation where just users should just be aware of what inputs they're providing into the tool and just make sure that there's not any duplicate information that they could encounter. We are we have already added a little bit of filtering and mitigations to catch this, and we've explained some more of this in the paper itself. But that's definitely, I think, that probably the trickiest part.
Yeah, and I kind of wanted to add, like, lightly, like, one of the things that we actually encountered with this project is that science, when you're, I guess, like a kid or like an undergrad, you think of science as this very like neat idea, everything's like very well organized. And then the truth is, when you start tackling certain problems, you realize that a lot of the, I guess, like unexplored or cutting edge areas are still being developed. And so there's not like a standard, or some people don't really follow the standard if there is one, because it's not universal. And so what we would find is that you search in a database and there's a lot of dirty data. Well, I mean, let's call it dirty data, where they're not labeled correctly, or they're labeled in a way that you can't process. You have to specifically tackle that. And so, I guess one of the critical steps is like Sriram's saying, is like understanding the data, like why there might be differences between doing one search versus another, versus like searching in another database directly. So yeah, it's one of the critical things about using our tool is truly to understand that there's some limitations, right? To how much you can process, and the results might not match between one database and another, or searching in that same database in the web page itself. But yeah.
Yeah, actually, that also just reminded me. We do recommend generally that users start with the most restrictive types of search and then see what they can find and then loosen up from there. It's the classic bias variance trade-off, I guess. But because there are some of these cases where the databases may not have perfectly well annotated data, you want to start by using restrictive searches so that you know what you're getting is high quality, and then maybe loosen up from there.
Yeah.
I mean, you developed this tool, like you had the idea, you went on, you developed a tool, but then you took a step further and sent it to peer review. What was the motivation to go that far and have your tool and your protocol peer reviewed and published as a standalone methodological paper?
Yeah, so when I guess like when we started, we didn't have a clear idea, like, this was gonna be like something we wanted to publish. But the more that we interviewed scientists and kind of as we started working and kept developing the tool, we realized that one of the most important things about science, I guess, and what makes science is not only like reproducibility, but also like the idea of peer reviewing, making sure that other people from that have nothing to do with you can look at your paper and say this is good work. And so we always kind of wanted a way to validate our tool, to have like people judge what we wrote, how we did things, and then like approve it, or give us feedback if they thought something was needed improvement or whatnot. And so PLOS ONE was a kind of like a very nice kind of paper to publish on, because they have this protocol attached. And so we can kind of both publish a paper that talks about, why this tool is important, the biological aspect of it, some use case or theoretical use cases where this tool can be applied. And then also have like a protocol that kind of speaks to how to actually use this tool. What are the steps? What are the, how to make it easy to replicate what we're doing, right? And to have all this peer reviewed, I think it was very important to make sure that it works, and also to have scientists trust that what we built is good.
How did the feedback that you received from the peer reviewers shape the final version of their protocol? And also how you guys as undergrads navigated that process and the dialogue with the peer reviewers?
Yeah, sorry, when you were just asking that question, I had thought about another point I wanted to mention about the previous point about why we decided to publish. Um, the other thing to mention was that also PLOS ONE is not pay to read. And I think a large part of the point of our tool is accessibility. There are other tools that query for reference sequences out there, but they require some amount of coding expertise, whereas ours is we have a GUI. We don't need any of that software background. And there's a lot of users who do want to know about whether, reference sequences are available, what the scope of an eDNA project might be, like land managers who aren't trained in computer science at all, and community scientists, for example. Um, and so we wanted to make sure that it was just accessible. So that was another big motivation. As far as the peer reviewers, I think we sort of touched on that earlier, is that it was um, a good chance to like really have a formal process of sitting down and have somebody reviewing our work and thinking about aspects of this field that we might not have catched or that we might not have caught. But also when we started working on this project, this was, a few years ago, and there were fewer options available, and there were like fewer other such tools that are doing something similar to what we're doing. Um, and we hadn't we didn't really think of making a comparison at that point in time, because when we were first working on this project, when we were first doing all these interviews with the various scientists, um, everything else was like sort of new. Everything was sort of first of its kind. So there wasn't really so much comparison points in the papers that we had seen. But the reviewers did bring up, hey, this would be something really important to add to the paper, which we really appreciated.
Yeah, we definitely like it shaped the way that like there's a when we wrote the paper, yeah, like as Sri said, we didn't really compare it to many of the things out there. And so when the peer reviewers brought this up, we decided to actually go take it one step further and not only make like a kind of like a small section where we kind of thoroughly went through what is out there, what is different about it, and some of the pros and cons. And so yeah, it definitely shaped a fair amount of the paper. Also, to clarify some points, right? Some of the peer reviewers read what we said, and they pointed out, some of the stuff is not clear, the way that you talk about this table or this description here, and so it helped us improve the paper, slightly, and explain things better and whatnot. So it was definitely very helpful. And actually, just to tackle, I think you mentioned like how it was that we handled, since we're students, it was very new. And yeah, actually, we were a bit unsure at first because it was the first time that we were, going through this process on our own. But it actually was quite easy, I have to say. Like it's a little daunting because like you don't know anything, and it's this very serious process of science, peer reviewing. But then in the end, it was actually very nice. We sent a paper, we got some feedback back, and the feedback was very nice. It was well organized, and we could read it. It didn't feel like scientists or other people were like being like really rude. They were pointing out things that the paper had flaws in, and then we answered to each of those points. We went one by one, and we improved our paper and then sent it back, and it was quite simple actually.
And why did you decide to make the peer review history public together with your paper?
Yeah, I mean, like we said, I think that this was a case of like this was our first time with the peer review process for sure. And I think that there was number one, I mean, like science is all about, transparency, right? And making sure that the process is clean and thorough. And we felt like it just on principle, it made sense to do that. But also further than that, because we are students who are not necessarily working with a lab or another like mentor, we thought that perhaps if there was some way that like other people heard this story, that they might be somewhat they could like at least look at uh our peer review and know a bit more about what the process is like, right? If they're in a similar situation to us and they don't have that kind of they don't know what they're walking into, right? It's nice to have something out there.
So the next question is my last question, and it might be speculative at this time because the paper is relatively young. I think it was published about a month and a half ago or so. But what do you believe, or what do you know it has been or will be the impact of publishing this paper for both of your careers, considering you are like really young and just taking off?
Yeah, so I mean, I think what will be the impact of the paper itself? I mean, one can always hope. I hope that, you know, it's used by many of the scientists that we talked to. And, you know, I hope that this paper kind of brings light into, you know, that, you know, we were very serious about this tool. We were, you know, we worked very hard for it. And so I'm hoping that, you know, it will shed light into this tool and, you know, scientists out there will use it. But you know, regardless of you know, whether the paper is, you know, very successful and a lot of people cite it, which I hope so, it actually helped us on a personal level a lot because, for example, for me, it shaped what I was gonna do after undergrad. You know, when I was an undergraduate, um, and I started on this group, it was kind of my first attempt at research, and not only just research, it was research on our own, and so it was, you know, it helped. Kind of visualize what that process is like. It wasn't with a lab, there was no formal PI, but you know, we were still solving problems that, like, you know, nobody had solved, really, trying to make an app that, you know, was very usable. And then finding new problems, like, you know, every time we try to implement something, something breaks or goes wrong, and so you have to come up with ways to solve them. And so I think that motivated me to, or well, it showed me that I really liked that process. I really enjoyed that challenge. I really enjoyed that research. And so I decided to go to grad school, and I applied for a master's, and I got into Cornell, and now I'm doing my PhD. So it definitely shaped the way I went about things. I have shifted my you know, work. I used to be, I guess, biology and computational biology. Nowadays, I'm more in like the medical field. But still, it was a very you know, eye-opening experience, and it showed me what, you know, research is like, and it was a great life-changing experience, I guess, in a way.
Yeah, I mean, on my end, all of the same learnings apply. There was a ton of amazing soft skills in the case of, like, this was the first truly open-ended problem-solving project I have been on, because usually in school it's like everything's put into clear bounds, right? Whereas this was a case where there was like no obvious clear solution. And I found that I really like having that agency in the problems that I work on, and also um, just yeah, like I like the small groups, the startup kind of feeling. And aside from like all of both the technical and soft skill learnings that I had, I mean, I want to keep doing more of this in the future. I'm taking some time to sort of like buff up my technical skills, but later I want to really pursue that intersection between that computational biology intersection between like these environmental sciences and the computer science theory that I already have a foundation in.
I mean, thank you so much, guys. It's been like really, really inspiring talking to you, like so young, so motivated, so clever and goal directed. So it's been really a pleasure. I don't have any more questions. I don't know if you want to add anything else that haven't been discussed so far.
No, really. I mean, thank you so much for everything. I mean, like, for inviting us. It was great talking about this process. I know it's very weird because, like, you know, most papers, they don't, you know, they have the author list is, you know, has a formatting and everything, and we were kind of like, as a student, it's very daunting. You don't really fill all the gaps. But I guess if I'm gonna finish with this with some, you know, with like a note, if you're a student and, you know, you want to do your own research, I think go for it. Scientists are actually very open to working and like interviewing. People out there are actually a lot more willing to talk to you and like help you in certain things that you might expect. So if you're a student out there and, you know, you want to do something, I would say pursue it. But yeah, thank you so much.